Hello, my name is Dalton Lucas, and I'm a first year PhD student in Dr. McFarland's lab. And today I'll be presenting our group's research on the synthesis and characterization of light-triggered metal complexes for cancer treatment. So, a brief introduction is we employ photodynamic therapy along with the photosensitizer and a specific wavelength of light to then generate reactive oxygen species within the individual's body to selectively target cancer cells and sparing the healthy tissue, which is a big contrast to conventional cancer treatments such as chemotherapy, where it has pretty severe and systemic symptoms. So photodynamic therapy is going to be where an individual is going to receive a drug, which is this case going to be a photosensitizer, and within 1 to 2 days or 1 to 3 days, this photosensitizer is going to be absorbed in the individual's body and within the cancer cells, and along with the help with MRI, the individual is going to be treated with a specific wavelength of light to then induce reactive oxygen species and singlet oxygen to then destroy the tumors and induce an anti-tumor immune response, and then hopefully kills all the cancer cells. So the objectives of the study was to synthesize, characterize, and evaluate a new class of ruthenium terpyridine oligothiophene complexes, and then to determine the effect of the oligothiophene chain on the biological activity. So we have our two target structures down here, bis homoleptic ruthenium terpyridine, along with the ruthenium terpyridine with the varying oligothiophene chains. So for synthesis, we have our terpyridine ruthenium, and we react that with ruthenium trichloride and water, the following conditions, and then in situ, we add our corresponding terpyridine with the varying thiophene chains, which would then generate our Ru1T or Ru4T, depending on the number of thiophene units. And then to synthesize the bis homoleptic ruthenium terpyridine, we did a 2-to-1 stoichiometric ratio of the terpyridine to the ruthenium trichloride to synthesize that. So compounds are purified with CombiFlash column chromatography with the following gradient with 0.5% KNO3, 7.5% water, and acetonitrile. Those could be converted to PF6 salts for EChem studies with saturated KPF6 and converted back to chloride salts using amberlite ion exchange chromatography. And Cephedex can be used to further purify the compounds, and purity was obtained greater than 95% and confirmed by HPLC. For characterization, we have proton NMR, 2D correlation spectroscopy, as well as high resolution ESI positive mass spec. And fluorine NMR was also performed to confirm that when we had the chloride salts, there was no more fluorine left. And for the photobiological evaluation, these are done on a melanoma cancer cell line, and both in hypoxia and normoxia conditions. And what we see here is Ru2T is an outstanding outlier, and this goes in contrast with what we previously studied on our ruthenium polypyridal complexes, where as you increase the number of the oligothiophene chain, you will see a corresponding increase in biological activity. So this is pretty interesting observation here. And in the future, we do plan to increase the number of biological replicates and to introduce some EChem studies as well into this work. But to conclude, we synthesized a new ruthenium-2 terpyridine complex family. We characterized them and also evaluated their photobiological efficacy against cancer cells. And as I mentioned earlier, the Ru2T showed the highest potency in normoxia conditions. And I'd like to thank Dr. McFarland and the rest of my colleagues, since this is a multidisciplinary approach and research project. Thank you very much.